topic. So let the recording commence. Yeah. So good morning, dear students. So we'll commence with the topic of neural control and coordination. So the coordination that is involved in the functioning of the nervous system. You might have studied in your lower classes the nervous system. It is of different type: CNS, PNS, ANS. That is the central nervous system, which consists of the brain and spinal cord. So brain is protected within the cranium. So the that is the central nervous system. Then peripheral nervous system. So based on their functions, you might have studied about the sensory nerves, motor nerves, and the mixed nerves. So some of these nerves which are both sensory and motor in nature, we call them as mixed nerves, or based on their path of exit or entry. So we had the afferent nerves and efferent nerves. So they are the peripheral nervous system. The autonomous nervous system. So two types you might have studied about. So the sympathetic and parasympathetic nervous system. So under the autonomous nervous system, you might have studied about the sympathetic and parasympathetic nervous system. So in the backdrop of this uh, slide, you can see the nervous system is focused. So the spinal cord, the medulla oblongata, it is going to proceed as spinal cord, so which is passing through the neural column of the vertebra. So the vertebral column has a opening called as the neural point or neural opening. So the spinal cord passes through them. Okay, so you have 12 pair of we have 12 pair of cranial nerves, and the spinal nerves are also there. So we'll be studying in detail about this nervous system and the neurotransmission and disorders of the nervous system. Okay. So the neural system, it is a system or nervous system, we also call it as. It is a system that controls and coordinates the body activities, conducts and integrates the information, and respond to stimuli. So now we had an example. My audio was not working. So that was the stimulus. So immediately you all responded, isn't it, that the audio isn't working. Okay, so here you are able to sense that it is a system that controls, the nervous system is a system that controls and coordinates the body activities. So you were listening to it. That was the response. For the stimuli was my talking was the stimuli for you, and you were listening to that. And that information enters into your brain and you remember them. There is a short-term memory, long-term memory. Some of it might have a deep impact. It has a long-term memory, and so the information is processed within your mind. See, all these are the activities of the nervous system. So neural system is a system that controls and coordinates the body activities. It conducts, the neural system conducts and integrates the information. So now you're integrating the information, whatever I'm saying, and respond to stimuli. So it conducts and integrates the information and respond to stimuli. That is being done by this nervous system. So the neural system includes brain, the spinal cord, which is passing through the neural opening of the vertebra, and the nerves from them. So the cranial nerves as well as the spinal nerves. So it is made up of specialized cells. So the uh, neural system or nervous system is made up of specialized cells known as neurons. So one more thing that you have to remember is neurons do not multiply. They remain the same from the infant stage to the grown-up adults. Only thing, they increase in size. So if neurons are damaged, they cannot be replaced because neurons do not undergo cell division. So you should understand that. So the nervous system is made up of specialized cells known as neurons. So the neuron or nerve cell. So the neuron is the structural and functional unit of nervous system. So the neurons are the structural and functional unit of nervous system. It has three main parts, that is the cell body or cyton, dendron, and axon. So the dendron receives the information from different source and they conduct the information to the cell body, and from the cyton, which we also call it as, from the cell body, the information is carried out, that is through axon. So neuron is also called as the nerve cell. It is a structural and functional unit of nervous system. It has three parts, that is the cell body, which is called as cyton, then the dendron and axon. We have multipolar neuron, so wherein we have many dendrites. They get the information towards the cyton, and that process information is processed and the response is sent through the axons. The impulses are carried away from cyton by the axon. Okay, so that we'll be studying about in the structure of multipolar neuron. So now just remember, the neuron or nerve cell is the structural and functional unit of the nervous system or neural system. It has three main parts: the cell body, which is also called as cyton, then they have dendrons and axons. So now if you study the structure of a multipolar neuron, as I told you, they have many dendrites. Okay, and this is the cyton. Cyton has a prominent nucleus, and they have this Niesel's granules within the cytoplasm of the neuron. See the membrane of the neuron, we call it as neurolemma. The cytoplasm of the neuron, we call it as neuroplasm. So the neuroplasm has numerous granules, and these prominent granules are nothing but tigroids. So they are also named as Niesel's granules or tigroids. So this is the cyton. So and they are the receiving many information from many dendrites. Dendrites are many, and they have a single axon. So here this we call it as axon hillock. So the axon is protected by this myelination. So they are also having the Schwann cells. In between the Schwann cells, the junction we call it as node of Rademacher. So Schwann cells are also having this nucleus. So the Schwann cells are uh, engulfing or covering the axon. So the axon branches, and the axon terminal has a small button-like structures which we call it as synaptic knobs. So the axon terminal, they end up with small button-like structures called as synaptic knobs. Okay, so the cell body or cyton, it contains cytoplasm, that is neuroplasm, cell organelles they also contain, and Niesel's granules, so which are granular bodies. So Niesel's granules are also called as tigroids. So the short fibers that project out of the cell body or cyton, so that is dendron. So the short fibers that project out of the cell body or cyton, we call it as dendrons. So sub branches of dendrons are called as dendrites. So the sub branches of this dendrons are called as dendrites. So the main branches we call it as dendron, the sub branches we call it as dendrites. So the dendrites, they transmit impulses towards the cell body or cyton. So they're getting the impulses towards the cell body or cyton. So the axon or nerve fiber, we call it as a long fiber which transmit impulses, a long fiber which transmit impulses away from the cell body. We call it as axon. A long fiber which carries impulses or which transmits impulses away from the cell body or cyton, we call it as axon. Axon is also called as nerve fiber. So the branching of axon is called axonite. So the branching we call it as axonite. Dendron, dendrite, axon, axonite. Okay, so each axonite ends as a bulb-like structures called as synaptic knobs. So the each axons, they end up with a button-like or bulb-like structure called as synaptic knobs. So the types of neurons that we have is the multipolar neuron. So the multipolar neuron has a single axon and two or more dendrons found in the cerebral cortex. So multipolar neuron is majorly noticed in the cerebral cortex. Okay. So the bipolar neuron, generally the motor neurons are multipolar neurons. Okay, so one axon it has, and two or more dendrons. So they are found in the cerebral cortex, the multipolar neuron. So the bipolar neuron, they have one axon. So the bipolar neuron has one axon and one dendron found in the retina of the eye. So the bipolar neuron has one axon and one dendron, and they are found in the bipolar, found in the retina of eye. So the bipolar neuron is found in the retina of eye. So the unipolar neuron, one axon only. So they have only a single axon, so they are found in the embryonic stage. So the unipolar neuron, they have a single axon and they are found in the embryonic stage. So the unipolar neuron. Okay. So this is what you have to remember regarding the types of neurons. So there are three types of neuron, the multipolar neuron. So they are found in the cerebral cortex. So they have a single axon and two or more dendrons. They are found in the cerebral cortex. Okay, the bipolar neuron, they have one axon and one dendron, so they are found in the retina of the eye. The unipolar neuron, they are having one axon only, so they are found in the embryonic stage. So they don't have any dendrons, they are found in the embryonic stage, the unipolar neuron. The axons are of two types, the myelinated axon and non-myelinated axons. So the axons are of two types, the myelinated axon and non-myelinated axon. So myelinated axon, it is enveloped with Schwann cells. The myelinated axons are enveloped with Schwann cells, which forms a myelin sheath around the axon. So the myelinated axon, it is enveloped with Schwann cells. So it is covered by the Schwann cells, which forms myelin sheath around the axon. So they form the myelin sheath around the axon. So this myelinated axon is found in spinal and cranial nerves. So the myelinated axon is found in the spinal and cranial nerves. So the white colored area formed of myelinated nerve fibers is called white matter. So, so myelinated axons, they would be generally enveloped with Schwann cells, which form a myelin sheath. So the Schwann cells form a myelin sheath around the axon. So I told you the node of junction in between the Schwann cells. So where are these myelinated axons found? They are found in spinal and cranial nerves. So the myelinated axons are found in the spinal and cranial nerves. So you can see the Schwann cells, and in between you have this node of Ranvier. So this is the Schwann cells. Okay. So the white colored area is formed of myelinated nerve fibers and it is called as white matter. So the white colored area is majorly formed of myelinated nerve fibers and they are called as white matter. So the gaps between two adjacent myelin sheaths or Schwann cells, we call it as nodes of Ranvier. So the gap between two adjacent myelin sheaths are called as nodes of Ranvier. So it is enveloped by Schwann cells which form a myelin sheath around the axon. So the gaps between two adjacent myelin sheaths, they are called as nodes of Ranvier. So the gap between two adjacent myelin sheaths are called as nodes of Ranvier. So the non-myelinated axons, they are the Schwann cells present but no myelin sheath. So Schwann cells are present, but there are no myelin sheaths. Okay, the gray colored area without myelin sheath is called gray matter. So it is found in autonomous and somatic neural systems. So the spinal and cranial nerves, you find the myelinated sheath, and the white matter is because of the myelinated sheath. The gray colored area without the myelinated sheath is called as gray matter. So they are found in the non-myelinated axon is found in autonomous and somatic neural systems. So the autonomous and somatic neural systems are having this non-myelinated axons. So myelinated axons. So you can here notice the gray matter and white matter. So white matter is interior, gray matter is exterior. So it is vice versa in spinal cord. So the gray matter is interior and white matter is outside. So whereas in brain, the gray matter is exterior, white matter is interior. So white matter is made up of this myelinated axons. Gray matter is made up of this gray colored area. They are made up of this non-myelinated axons. Okay. So it is enveloped with Schwann cells which form a myelin sheet around the axon, and they form a myelin sheet around the axon. So Schwann cells are present, but no myelin sheath. So the gray colored area without myelin sheath is called as gray matter, or non-myelinated axon is called as gray matter. So they are found in non-myelinated axon is found in autonomous and somatic neural systems, whereas the myelinated axon is found in spinal and cranial nerves. So non-myelinated is found in the autonomous nervous system and somatic neural systems. Next, we'll be studying about the generation and conduction of nerve impulses. So how is the nerve impulses generated and conducted? So that we are going to study about. So you can see in the backdrops, so the messages being carried, received, being sent through this nerve fibers. So the generation and conduction of nerve impulses. So impulse transmission is electrochemical in nature. So you can see this backdrop slide. So impulse transmission is electrochemical in nature. So three steps of impulse transmission are: first one is there is a maintenance of resting membrane potential. Second one is action potential, propagation of action potential. So you can remember it as MAP, MAP, MAP. That is first step in the impulse transmission is maintenance of resting membrane potential. The maintenance of resting membrane potential. Second step is the action potential. Third step is propagation of action potential. So you can remember it as MAP, maintenance of resting membrane potential, that is M. A is action potential. So P is propagation of action potential. So you can remember it as MAP. So first we'll understand about the uh, first step, that is maintenance of resting membrane potential. So the membrane, neural membrane, they contain various selectively permeable ion channels. So the neural membrane is also phosphate biphospholipid layer. So you have studied the structure of membrane, isn't it? Cell membrane or plasma membrane. So where they have this phosphobilipid layer with proteins, integrated proteins, surface proteins, peripheral proteins. We have studied about that. So here the membrane potential, resting membrane potential, that has to be maintained. How is it being done? So the neural membrane, they contain various selectively permeable ion channels. So they contain various selectively permeable ion channels. Okay. So in a resting neuron not conducting any impulse, the axonal membrane is in a resting neuron. So where the neuron is not conducting any impulse, the axonal membrane, the membrane of the axon, is more permeable to potassium ions and nearly impermeable to sodium ions. So in a resting stage, the axonal membrane is permeable to potassium ions and impermeable to sodium ions. Also, the membrane is impermeable to negatively charged proteins in axoplasm. So the fluid of the axon, we call it as axoplasm. So the membrane is impermeable to negatively charged proteins in the axoplasm. So this is what is the maintenance of resting membrane potential. So the neural membrane contain various ion selectively permeable ion channels. The neural membrane, they contain various selectively permeable ion channels. In a resting neuron, that is neuron which does not conduct any impulse, the axonal membrane is more permeable to potassium ions than and nearly impermeable to sodium ions. Also, the membrane is impermeable to negatively charged proteins in the axoplasm. So this is what is you have to remember regarding the maintenance of resting membrane potential, RMP, maintenance of resting membrane potential. So the concentration of potassium ion and negatively charged protein in axoplasm is high and concentration of sodium ion is low. So that is what you can notice in the resting membrane potential. So concentration of potassium ion and positively charged particles is high in the axoplasm than in the, and concentration of sodium ion is low. So you can see the concentration of potassium ion and sodium ion within the axoplasm. So the concentration of potassium ion is more, and the negatively charged uh, proteins in axoplasm is high, and concentration of sodium is low. The fluid outside the axon, so this is the fluid outside the axon. They contain low concentration of potassium ion and high concentration of sodium ion. So this is the axon, axoplasm, that's the inside part. The outside the fluid will be having potassium ions lesser and sodium ions will be higher. So this forms an ionic or concentration gradient across the resting membrane. So there is a difference in concentration. So sodium ions are higher outside and lower inside. The potassium ions are higher concentration is inside and lower outside. So this forms an ionic or concentration gradient across the resting membrane. So there is a concentration gradient across the resting membrane. So within the axoplasm, so the potassium ions, you can notice the units 400 so micromoles. So the potassium ions are more and the sodium ions are less, whereas outside in the fluid, so outside the membrane, the fluid that they have sodium ion are more and potassium ions are less. So there is a concentration gradient uh, between these two, between the inside and outside of the membrane. There is a concentration gradient. So this forms a concentration gradient across the resting membrane. Okay. So this is the axon. So the extracellular, intracellular within the axon, and again there is an extracellular. So you can notice that. So the ionic gradients are maintained by the active transport of ions. So by the sodium potassium pump. So the how is the ionic gradient maintained? So they are maintained by the active transport of ions by the sodium potassium pump. It transports three sodium ions outside and for two potassium ion into the cell. So it transports three sodium ions outside for every two potassium ions which enters into the cell. So three sodium ions goes outside for every two potassium ions that enter the cell. As a result, the outer surface becomes positively charged and inner surface becomes negatively charged. So that is polarized. So the outer surface becomes positively charged and the inner surface becomes negatively charged. So they are polarized now. So the electric potential difference across the resting plasma membrane is called as the resting potential. So the electric membrane difference across the resting plasma membrane is called as the resting potential. So what is resting potential? The electric potential difference across the membrane, across the resting plasma membrane, we call it as the resting potential. So still now we discussed about the first aspect of it, the impulses, conduction of impulses. So generation and conduction of nerve impulses, that is maintenance of the resting non-membrane potential. So they have this ionic channels so through which they are selectively permeable for during the resting stages. They are selectively permeable more for sodium ions, more for potassium ions whenever they are in rest, and they are almost impermeable to sodium ions. And they are also impermeable to the proteins. Okay, so as a result of that, there is a electric potential difference across the resting plasma membrane is called as the resting potential. So the resting plasma membrane has an electric potential difference. So for every 3 sodium ions that enters, that goes outwards, 2 potassium ions outwards for 2 potassium ions which enter inside. Okay, as a result, the outer surface becomes positively charged and the inner surface becomes negatively charged. So therefore, they are polarized. So the electric potential difference across the resting plasma membrane, it is called as the resting potential. Now, the next part that we are going to understand is the action potential. So when a stimulus is applied, the membrane at site A, they become permeable to sodium ion. So this causes usually in the resting potential, they are permeable to selectively permeable to potassium ions and almost impermeable to sodium ions. But when a stimulus is applied, the membrane at site A becomes permeable to sodium ion. This causes a rapid influx of sodium ion, entry of sodium ion, and reversal of the polarity at that site. So the outer becomes negative. So there is a reversal of polarity, isn't it? So the outer becomes negative and the inner becomes positive. It is called as depolarization. So the polarity changes this rapid. So whenever there is a stimulus is applied, the membrane at site A becomes permeable to sodium ion. This causes rapid influx of sodium ion and reversal of the polarity at that site. So the outer becomes negative and inner becomes positive at this site. Okay, so this is called as depolarization. So the electrical potential difference during depolarization. Okay, so the electrical potential difference, so across the resting plasma membrane, we call it as resting potential. The electrical potential difference during depolarization across the plasma membrane, we call it as action potential, and that is nothing but nerve impulse. This we call it as action potential. The third part of this is propagation of action potential. So at sites ahead, that is A site we have already discussed. So at sites ahead, that is site B, outer surface is positive, inner surface is negative. As a result, a current flows on the inner surface from A to site B. From A to site B, the current flows in the inner surface. So at site B, they are the outer surface would be positively charged and inner surface will be negatively charged. So as a result of it, the current flows in the inner surface from site A to site B. On the outer surface, current flows from site B to site A. So that is happening the vice versa. So from site B to site A. So on the outer surface, the current flows from site B to site A. Okay. So to complete the circuit, so here from the current flows from site A to site B. So current flows on the inner surface from site A to site B. So in the outer surface, the current flows from the site B to site A to complete the circuit. So the polarity is reversed and action potential is generated at site B. So now this polarity is reversed and action potential is now uh, action potential at is generated at site B. That is action potential at site A arrives at the action potential at site A, they arrive at site B. Okay. The sequence is repeated along the axon and the impulse is conducted. The rise in permeability to sodium, the rise in permeability to sodium ion is extremely short-lived. Okay, it is quickly followed by a rise in permeability to potassium ion. So the rise in permeability to sodium ion is very short-lived. It is immediately replaced by a rise in permeability to potassium ion. So the immediately potassium ion diffuses out, that is outside the membrane, and restores the resting membrane. Thus, the fiber becomes ready for further stimulation. So immediately the potassium ion diffuses outside the membrane and restores the resting membrane. So thus the fibers becomes ready for another stimulation. So this is how the stimulation is passed. So the generation and conduction of nerve impulses is flow of the electric impulses. Okay. So impulse transmission is electrochemical. So three steps of impulse transmission are there. First is maintenance of resting membrane potential. Whenever they are in resting membrane potential, the membrane, they have the ionic channels. So the membrane is selectively permeable to the potassium ions and almost impermeable to the sodium ions. So the protein, uh, so that is what we had, more permeable to potassium ions and nearly impermeable to sodium ions. Also, the membrane is impermeable to negatively charged proteins in axoplasm. So as a result of that, so concentration of potassium ion and negatively charged proteins in axoplasm is high and concentration of sodium ion is low. So the fluid outside the axon contains low concentration of, so you can notice here, low concentration of potassium ions and high concentration of sodium ions. So there is a formation of the concentration gradient or ionic gradient across this resting membrane. Then there was the ionic gradients are maintained by the active transport of the ions by the sodium potassium pump. So active transport of ions, it implies uh, so the expenditure of energy. So the ionic gradients are maintained by the active transport of ions by the sodium potassium pumps. So it transports three for every transport. So how is this being maintained? The potassium ion more towards the inside and sodium ion less towards the inside, potassium ion less towards the outside, and sodium ion more. How is this ionic gradient being maintained? So it is an active process, I've already told you, wherein, so for every transport of three sodium ions outwards, two potassium ions enter into the cell. For every transfer of three sodium ions outside, so there is entry of two potassium ions into the cells. As a result, the outer surface becomes positively charged and the inner surface becomes negatively charged. That is, they are polarized. So the electric potential difference across the resting plasma membrane, the electric potential difference across the resting plasma membrane, this is how it is. The electric potential difference across the resting plasma membrane, it is called as the resting potential. Okay, during action potential, whenever a stimulus is applied, the membrane at site A becomes more permeable to sodium ions. This causes rapid influx of sodium ion and reversal of polarity at that site. So you find that because of the entry of sodium ion inside becomes the positive and the outside becomes negative, and reversal of the polarity at the side. So outer negative and inner positive. So it is called as depolarization. During action potential, when a stimulus is applied, depolarization takes place by when the sodium ions become permeable. So it is called depolarization. The electric potential difference during depolarization across the plasma membrane, the electric potential difference during depolarization across this plasma membrane, we call it as action potential or nerve impulse. Action potential is nothing but nerve impulse. So how is the propagation of action potential or nerve impulse taking place. At sites ahead, that is site A, we know where they have received the stimulus and action potential has been generated. So the at site ahead, the outer surface is positive and inner surface is negative. As a result, a current flows on the inner surface. So there is a current flow in the inner surface from site A to site B, inner surface. On the outer surface, current flows from site B to site A. So there is on the outer surface, the current flows from site B to site A. On the inner surface, the current flows from site A to site B. So to complete the circuit here, the polarity is reversed and action potential is now generated at B site. That is action potential at site A. It arrives at site B. So this is how the impulse is being transmitted. So the propagation of action potential. So as a at site B ahead of it, outer surface is positive and inner surface is negative. As a result, the current flows on the inner surface from site A to site B and outer surface from site B to site A. So to complete the circuit, hence the polarity is reversed and action potential is generated, okay, at site B. So that is action potential at site A arrives to site B. The sequence is repeated to site C and others. So the sequence is repeated along the axon and the impulse is conducted. So the rise in permeability to sodium ion is extremely short-lived. Okay, it is quickly followed by rise in permeability to potassium ion. So the rise in permeability to sodium ion is very short-lived and it is quickly followed by a rise in permeability to potassium ion. So immediately potassium ion diffuses outside the membrane and restores the resting membrane. So thus the fiber becomes ready for further stimulation. So immediately potassium ion diffuses outside the membrane and restores the resting membrane. Thus the fiber becomes ready for further stimulation. So the next part that we are going to understand is the synaptic transmission of impulses. Synapse is a functional junction between two neurons. So synapse is a functional junction between two neurons. It is of two types: electrical synapse, chemical synapse. Synapse is the functional junction between two neurons. It is of two types: electrical synapse, chemical synapse. So in electrical synapse, the membranes of pre and post synaptic neurons. See the pre and post synaptic neurons, the membranes of presynaptic and prosynaptic neurons. So this is the presynaptic and this is the postsynaptic membranes. So in this, the membranes of presynaptic neurons and postsynaptic neurons so are in close proximity. So the impulse transmission is similar to the transmission along an axon. So from site A to site B, we just studied about the action potential. So the impulse transmission is similar to transmission along an axon. So in this, the membranes of presynaptic neuron and postsynaptic neurons are in close proximity. So the presynaptic neurons and the close and the postsynaptic neurons are in proximity. Okay, so impulse transmission is similar to the transmission along an axon. So impulse transmission is faster than in chemical synapse. So electrical synapse, the impulse transmission is faster than in chemical synapse. Electrical synapses are very rare in human system. So electrical systems are electrical synapse are impulse transmission is faster in electrical synapse than in chemical synapse. Electrical synapses are very rare in human system. So that is what we have to understand about. So the chemical system or chemical synapse, in this, there is a fluid filled space or synaptic cleft between the presynaptic neuron and postsynaptic neuron. So there is a fluid filled uh, space or synaptic cleft between the presynaptic neuron and the postsynaptic neuron. So there is a fluid filled space. So we call it a synaptic cleft. So between the presynaptic neuron and postsynaptic neuron. So the presynaptic neurons have swellings called synaptic knobs or buttons, synaptic buttons, we call it as. So they contain synaptic vesicles. You can see the synaptic vesicles filled with neurotransmitters. So the neurotransmitters are acetylcholine or adrenaline. So the presynaptic regions have swellings called as synaptic knobs, so or buttons. The synaptic knob contains synaptic vesicles which are filled with acetylcholine, that is neurotransmitters. They are filled with neurotransmitters, either acetylcholine or adrenaline. So impulse reaches the axon terminal. Okay, so the synaptic vesicles, so the axon terminal synaptic vesicles bind on the plasma membrane. So the release of neurotransmitter takes place. So impulses reach the axon terminal, synaptic vesicles bind on the plasma membrane. The release of neurotransmitter is released. It diffuses across the synaptic cleft, combines with the receptors of the post. So they combine with the neurotransmitter are released. They are combining with the receptors of the post synaptic membrane. Okay, so neurosynaptic, the postsynaptic membrane, you can see that they are going to combine with the receptors of the postsynaptic membrane. So the neurotransmitters are released. So opening of ion channels, allowing entry of ions, and it generates. So there is an opening of ion channels and there is an uh, allowing entry of ions. It generates an action potential. The action potential may be excitatory or inhibitory. So these are the steps that are taking place in chemicals. So the impulses are received at the axon terminal. So they bind on the once it has reached the axon terminal, the synaptic vesicles bind on the plasma membrane. So the release of neurotransmitters like acetylcholine or the adrenaline takes place. So this neurotransmitters that are released combine with the uh, receptors on the postsynaptic neuron. They combine with the receptors on the postsynaptic neuron. See, they have enlarged this portion and shown it here to you. So, and they are combining with the postsynaptic membrane. So the opening of the ion channels, allowing entry of ions. So it generates an action potential. So the action potential might be excitatory or it might be inhibitory. It might either excite or prevent. So this they have enlarged to show you how the neurotransmitters are entering through the synaptic cleft into the receptors of the postsynaptic membranes. So postsynaptic neurotransmitter receptors. Okay, the opening of this ion channels allow the entry of ions, that is acetylcholine or adrenaline, and they generate an action potential. Once the neurotransmitters enter this postsynaptic membrane, so they are going to generate action potential. The action potential might be either excitatory or inhibitory. Okay. After the completion of the different types of synapse we had studied about, so the electrical synapse and chemical synapse. So electrical synapse is very rare in humans. So chemical synapse is frequently found in human beings with the neurotransmitters. So the next that we are going to discuss about is the human neural system or human nervous system. So you can find the nervous system being highlighted here. So the brain, the spinal cord, the cranial nerves, spinal nerves, okay, the motor, sensory nerves, mixed nerves, all these are found within our human neural system. See, each and every part of the body is receiving the uh, nervous system. So there is a stimulus being conducted to the central nervous system. Okay. So the division of human neural system, I've already discussed about this. So the human neural system or nervous system, they consist of central nervous system, peripheral nervous system. The central nervous system consists of brain and spinal cord. So the peripheral nervous system consists of somatic neural nervous system and autonomic neural system. So this is another classification. So wherein the autonomic nervous system is placed under this peripheral neural system. So somatic neural system, SNS, ANS. Okay, the peripheral nervous system is into somatic nervous system and autonomic nervous system. So let us discuss about the brain. So brain, it is protected in cranial cavity or skull box. We have that which protects the brain. So it has three layers. So it has three layered connective tissue membranes. So they have three layer connective tissue membrane. So called as cranial meninges. So they have three layered connective tissue membrane called as cranial meninges. So you can see the outer dura mater, the middle arachnoid, and inner pia mater. So the outer dura mater, the middle arachnoid, so that the three layers of meninges. So it has three layered connective tissue membrane called as meninges. So you can see the cranial meninges. So there are three layers of cranial meninges. DAPA, you can remember DAP. Okay, the outer dura mater, middle arachnoid matter, and inner pia matter. So there is a subarachnoid space uh, between pia matter and arachnoid matter. So the subarachnoid space is there between pia matter and arachnoid matter, and it is filled with cerebrospinal fluid. So the subarachnoid space is filled with cerebrospinal fluid. So the ventricles of the brain are also filled with the cerebrospinal fluid. So the subarachnoid space between pia mater and the arachnoid is filled with cerebrospinal fluid. The ventricles of brain is also filled with cerebrospinal fluid. Brain has been, for our convenience, we have divided them into three divisions. The forebrain, which where we include the cerebrum, thalamus, and hypothalamus. The midbrain, uh, so the mesencephalon, forebrain is called as prosencephalon, midbrain is called as mesencephalon, hindbrain we call it as rhombencephalon. So the uh, midbrain and hindbrain. So the hindbrain, what does it consist of? It consists of pons, medulla oblongata, and cerebellum. So all three together we call it as hindbrain. So the forebrain, it consists of cerebrum, thalamus, hypothalamus. So all three together we call it as forebrain. The cerebrum, the cerebral cortex, you can notice cerebrum, the thalamus, hypothalamus. So we call them as forebrain or prosencephalon. So midbrain is called as mesencephalon. Hindbrain is called as rhombencephalon. They consist of pons, medulla oblongata, cerebellum. The next is forebrain. Now just look into the diagram. It is the anterior part of the brain. It consists of cerebrum, thalamus, and hypothalamus. So here we have, as per your textbook, it is the forebrain is of two types, cerebrum and diencephalon. So diencephalon. So they consist of thalamus and hypothalamus. Diencephalon consists of thalamus and hypothalamus. The cerebrum is the largest part. Okay, it has two cerebral hemispheres. So you have the left hemisphere and right hemispheres. So the cerebrum is the largest part. It has two cerebral hemispheres, that is right hemisphere and left hemispheres, so which are held together by a tract of nerve fibers, so which we call it as corpus callosum. So the left hemisphere and right hemispheres are held together. So you can see this, it is zoomed up. So they are held together by a tract of nerve fibers, which we call it as corpus callosum. So this is the cerebral hemispheres. There is a right cerebral hemisphere, left cerebral hemispheres. They are held together by a tract of nerve fibers, which we call it as corpus callosum. So the two cerebral hemispheres are held together by a tract of nerve fibers called as corpus callosum. The cerebrum will be continuing with it. So the outer part of cerebrum is called as cerebral cortex. It has convulsions and depressions and is formed of gray matter. The outer part of the cerebrum is called as the cerebral cortex. So it has convulsions and depressions, and this convulsions and depressions, it is formed of, see the sagittate section of brain, they might ask you for 5 marks. So you should be thorough with this diagram. So the it has convulsions and depressants, and it is formed of gray matter. So the outer part of the brain is a cerebral cortex. It consists of convulsions and depressions, and it is made up of gray matter. Okay, gray color is due to the presence of neuron cell bodies, so which are not myelinated. Inner part is formed of white matter where there is a myelinated fibers. So the gray color is due to the presence of neuron cell bodies. Okay, that is cytons. So gray color is because of the presence of neuron cell bodies. Inner part of the brain is formed of white matter. So the cerebral cortex consists of our forebrain, the prosencephalon, the cerebrum. They have different zones. Cerebral cortex consists of motor area. Okay, so controls voluntary movement of muscles. So you can notice this motor area, secondary motor area, premotor area. So this is the motor area. So the brain consists of the motor area, so which controls voluntary movement of muscles. Then they also consist of the sensory area. So the uh, next, the somatosensory. So you can notice the sensory or somasthetic area. So there is the sensory cerebral consists of the sensory or somasthetic area. They control the functioning of sense organs. Then association area. So it is neither clearly sensory nor is it motor in function. So they are responsible for intersensory association. So the cerebral cortex also has this association area. So which is neither clearly sensory nor motor in function. They are responsible for the various areas you can see. So they are responsible for intercessory sensory association, memory, and communication. So you can see this spatial association area, verbal association area. So there are various such association areas. It is neither clearly sensory nor motor in function. It is responsible, this association area is responsible for intersensory association, memory, and communication. So cerebrum, it is the integrated activities. So of different centers of cerebral cortex control, like intelligence, memory, judgment, learning, thinking, and articulate speech. So the integrated activities of different centers of cerebral cortex, they control intelligence, memory, judgment, learning, thinking, and articulate speech. All of these are associated with this cerebrum. So the integrated activities of different centers of cerebral cortex, they control. So you have the motor area centered, then there is a sensory area, association area, which is neither clearly nor sensory nor motor. Okay, the motor area controls voluntary movement of muscles. Sensory area, they control the functioning of sense organ. Association area, it is neither clearly sensory nor motor in function, but it is responsible for intersensory communication or association, memory, and communication. So the cerebrum has integrated activities of different centers of central cerebral cortex. They control intelligence, memory, judgment, learning, thinking, and articulate speech. Learning, thinking, and articulate speech. So forebrain is the prosencephalon. So already we have discussed about the cerebrum. The next is diencephalon. We'll be discussing two parts of it, thalamus and hypothalamus. So there is a right thalamus and left thalamus. So you can notice the right thalamus and left thalamus. So below the thalamus is the hypothalamus. Okay. So thalamus, it is the structure around which the cerebrum wraps. Okay, the structure around the thalamus is the structure around which the cerebrum wraps. So the cerebrum wraps around this thalamus. It is a coordinating center. So this particularly this thalamus is the coordinating center. So it is also called as relay station for sensory and motor impulses. So it is the structure. So thalamus is the structure around which the cerebrum wraps. It is a coordinating. There are left thalamus, right thalamus. You can see that it is a coordinating center. It relays. It is a relay station for sensory and motor impulses. So coordinating center or a relay station for sensory and motor impulses. Hypothalamus is seen below the thalamus. It function of this hypothalamus. Okay, so it is regulating the temperature, thirst, then hunger and emotions. So they secrete hypothalamic hormones. They control pituitary gland, control sleep, wakefulness, blood pressure, and heart rate. So the hypothalamus is seen below the thalamus. Okay, the hypothalamus is seen below the thalamus. So it regulates temperature, thirst, hunger, and emotions. So the hypothalamus regulates temperature, thirst, hunger, and emotions. They secrete hypothalamic hormones. They control the pituitary gland. Hypothalamus controls the pituitary gland. Hypothalamus also controls the sleep, wakefulness, blood pressure, and heart rate. So the hypothalamus also contains sleep, wakefulness, blood pressure, and heart rate. So the inner parts of cerebral hemispheres and a group of associated deep structures, okay, uh, like amygdala, hippocampus. So the amygdala, hippocampus, amygdala. So they are the inner parts of cerebral hemispheres and a group of associated deep structures like amygdala, hippocampus, hypothalamus, so etc. They together constitute the limbic lobe. So amygdala, hippocampus, and hypothalamus, etc., they constitute the limbic system or limbic lobe. It regulates, so these part of it, it regulates sexual behavior, motivations, emotions. So amygdala, hypothalamus, and hippocampus and hypothalamus, they constitute the limbic system or limbic lobes, and they are going to regulate the sexual behavior, motivations, emotions, excitement. The emotions can be excitement, pleasure, rage, fear. So they are all regulated by this limbic system or the limbic lobe. So the midbrain, so we'll be discussing it in the next class. It would be too much for you that at present. Okay, it is, I think, hindbrain we'll discuss in the next class. Midbrain, I'll complete it off. See, the midbrain is also called as mesencephalon. So it is located between, so the midbrain is located between the thalamus, hypothalamus, and pons. So the midbrain is located between the thalamus or hypothalamus and pons. So that we call it as midbrain. Okay, a canal, cerebral aqueduct, passes through the midbrain. So you can see the cerebral aqueduct, a canal, passes through this midbrain. So midbrain consists of four round lobes called corpora quadrigemina. They consist of four round lobes. Midbrain consists of, this is the region of midbrain, so through which there is a cerebral aqueduct which passes through the midbrain. So the cerebral aqueduct passes through the midbrain. So they are having four lobes. They consist of four lobes. So they are called as corpora quadrigemina. Their anterior pair is the center of visual reflexes, and the posterior pair, the corpora quadrigemina posterior pair, is a center of auditory reflex. So the midbrain is located between thalamus, hypothalamus, and pons. Okay, they are located between thalamus, hypothalamus, and pons. So the midbrain, so consists of a canal passes through the midbrain, that is the cerebral aqueduct. A canal passes through the midbrain called as cerebral aqueduct. So the midbrain consists of four lobes. Okay, so we call that as corpora quadrigemina. So their anterior pair is the center of visual reflexes, and the posterior pair is a center of auditory reflexes. So the anterior pair is it has a reflex of, okay, anyway, I was just looking for the diagram for corpora quadrigemina. That isn't there, doesn't matter. So the anterior pair of this corpora quadrigemina is a center of visual reflexes, and the posterior pair is a center for auditory reflexes. So that is what is about midbrain. So the hindbrain, we'll be discussing it in the next class. Okay. So any doubts or clarification, dear students? So in today's class, we had discussed about the nerve impulses and their transmission and types of synapse, that is electrical synapse and chemical synapse. In human beings, it is through chemical synapse. The neurotransmitters like acetylcholine and adrenaline. Okay, so these are the things that we have discussed in today's class. So do you have any doubts or clarification, dear students?